Okay. In case of fruit juice production, it's not like we use the juice. It's not like we create the juice. We make juice in a fermenter. No, 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 no. We basically produce some enzymes. We basically produce some enzymes in the fermenter, or correctly saying bioreactor. We produce some enzymes in the bioreactor that are going to help us in the juice production, and that enzyme is pectinase. A pectinase basically breaks down the pectin. Why it is important to break down the pectin? If you ever have treated the fruits for the juice, you would know that uh, a fruit juice, the fruit juice is like always have a shady appearance, a cloudy appearance, especially when it comes to the apple juice or maybe the orange juice. These kinds of juices, they have a cloudy appearance. So that cloudy appearance is because of the pectin, and it changes the original taste, maybe a bit the smell and some other things of the juice. So what do? We treat the juices with the pectin. We treat the juices with the pectin, but how are we obtaining the pectin? We are obtaining the pectin through the same phenomena. How we produce, or no, we basically use the fungus in the bioreactors, Aspergillus niger, and they produce the pectinase for us inside the bioreactor. So we just isolate that pectinase, uh, just apply it to the juice, and then when we filter the juice, the whole juice is going to be clear. There would be no cloudy appearance. Why? The pectinase breaks down the pectin to make the juice clearer. Okay. The next thing is biological washing powder. Now, what is the difference between an ordinary washing powder and the biological washing powder? Remember that a biological washing powder would contain enzymes. If you ever see that, if you ever have experienced, maybe you would have experienced this. Have you ever experienced the smell coming from a site where the milk was spilled?
Yeah, it doesn't smell nice.
That smell almost never gone.
Yeah.
Never goes. It never goes. Why it never goes? The thing is that the milk contains the proteins. You can wash with the help of the detergents. You can wash the milk fats. You can wash with the sugars, but you cannot wash the proteins. So what happens, that is the smell of the remainings of the milk that are under the process of decomposition. So if you apply the washing powders containing proteinases and lipases into it, you can very efficiently remove the stains from the clothes or from the different objects. The only problem is that if the washing powder contains a fabric dissolving enzyme, then it would take away your clothes as well. It would take away your clothes as well if it has fabric dissolving enzyme. But mostly biological washing powders, obviously if they are washing powders, they can never have a fabric dissolving enzyme. So the proteinases and the lipases are added into it, and when we mix, now how do we add them? That is the real question. See, most of the time the washing powder has the powdered form of detergent, but we have a very efficient technique. And what is that? Let me tell you. See, what happens that we have learned a technique, and you will experience this technique, um, literally experience this technique, because it is a practical in A levels biology. If you opt the biology for A levels, There is a technique called as immobilized enzymes. And immobilized enzymes are those in which, what do? We have very small beads made of different type of metals, and we place the enzymes onto it. The yellow ones are basically the enzymes that are permanently attached to the bead. And this bead, most of the time, it is made of jelly, agarose gel, or some other gel it is made of. So enzymes are permanently attached to the bead. Now, when the enzymes are permanently attached to the bead, now you would add them into the washing powder. And when you would add them in the washing powders, they can easily digest any particular lipid or lipid, fat, or protein like thing. Okay. Then comes the lactose free milk. Basically, we obtain the lactase with the same technology. The technology is not different. We grow the organisms into the bioreactors. The organisms make the lactase for us, and we use them. But remember that we are talking about the immobilized enzyme technique more in the lactose free milk. We obviously we The country in which you are living, the people are really conscious about any disorders there in a person, any disorders a person is born with. So they are extremely conscious about it. So there is a very high proportion of people who are really lactose intolerant. But obviously that that only comes to the surface when you really are looking for something. If you are not looking for something, it would not come to the surface. So that is why it seems that there's a very high proportion of people having the lactose intolerance there. So the lactose basically is not digested because the people cannot produce the lactase. So the lactose stays in the gut where it's fermented by the bacteria. Then there is like flatulence there, diarrhea, stomach pain, different problem. The lactase can be produced by on a large scale by fermenting yeast. Chloeveromyces fragilis. Cluveromyces fragilis, or fungi Aspergillus niger. Now these both fungi produce the lactase for us. They produce the lactase for us, and when they produce the lactase for us, we isolate the lactase and we immobilize it. A simple way is just add the lactase into the milk. The other way, a large scale method, is to immobilize the lactase. And how would it look like? See, the lactase would be immobilized like this. Hold on. Let's suppose you have a vessel. You have many beads in it. All the beads are containing the lactase enzyme. All of them are containing the lactase enzyme. And when you add the milk, when you add the milk, now this milk is containing the lactose. The lactose passes through the beads, getting digested. The milk passes through the bead, getting digested, and the milk that you are getting as a product is a lactose free milk. And remember, this is done at a larger scale. So we can easily do this procedure with the help of bioreactors. and then immobilizing the enzyme. See, two different techniques are under consideration here. Another thing, another thing that we can perform in the bioreactor is producing the antibiotics. Now, antibiotics basically were originated from the living sources. In other words, what I want to say is that antibiotics were originally discovered when the bacteria and fungi used to fight against each other. Obviously, they still do. So they fight against each other, and we extract the chemicals that are secreted by them. Do you know the experience of the Alexander Fleming?
No.
Alexander Fleming was the scientist who discovered the very first antibiotic, penicillin. And he was basically, it was the time of World War II when he was working on it, maybe World War One. And basically, the different kinds of soldiers were getting wounded. And when the wound was getting infected, they were having they were getting infected by a bacterium that was very, you can say, very much resistant to most of the antibiotics at that time, most of the chemicals at that time, because antibiotics, the term antibiotics was not known at that time. So what happened that Alexander Fleming was working on on the discovery of antibiotics, and he observed that what happened, that upon when he grew the same bacteria in the lab, when he grew the same bacteria in the lab, he saw that a strange type of a strange type of um, you can say mesh grew over the a strange type of mesh grew over the bacteria, and this mesh immediately started dissolving the bacteria, started destroying the bacteria, which basically means that at that time when he was working in the lab, the fungus came, a fungal spore came, and it created a mesh over the bacteria, started engulfing the bacteria. And when the fungus started engulfing the bacteria, first it was digesting it. Why? How it was digesting it? By releasing penicillin. So this is how the very first antibiotic was discovered. Although it is it is said that the penicillin was not useful for long because that particular bacteria got resistant again. So next, now the bacteria, the bacteria or fungi, they produce different kinds of antibiotics. They produce different kinds of antibiotics, and we isolate them. So this is the thing that is written over here, penicillin. Okay. Now what happens that this is commercial production of insulin. I'll not discuss it just yet. We first need to know how the insulin is going to be produced. First, we genetically modify the bacteria, then we start production. So first, we'll talk about the penicillin. Okay, the tanks are filled with nutrient solution. For penicillin production, the carbohydrate source is sugar, mainly lactose or corn steep liquor, and then contains amino acids as well as sugars. This is just the recipe of the culture of the nutrient solution. Remember, if you want the fungus to grow very efficiently there, you need to prepare the nutrient solution so that they can grow them as faster as they can. Then mineral salts are also there, and pH is adjusted, so you can feel that this is the recipe. It's just like They are telling you that keep the temperature at this, uh, keep these kinds of seasonings with you, salts, cardamom, this, and prepare the meat and rice, and you can make the famous dish biryani. It's just like that. So nutrient solution is just like the preparing the nutrient solution is just like preparing the recipe for the fermentation. Oh, sorry, for the penicillin production. A culture of appropriate microorganism is added to the nutrient liquid. Now, this is important. They are not telling us what kind of organisms. They are saying appropriate microorganisms. Remember that the antibiotics cannot be made without interaction of two different kinds of organisms, like the fungus and the bacteria. So remember that the fungus and the bacteria, they interact inside the fermenter, and the fungus produces the antibiotic, which we later isolate. You can see that if other bacteria or fungi get into the system, they can completely upset the process. And in the end, we filter it. You can see in this bioreactor, we are adding the feedstock, and all the antibiotic has been filtered. Now the organisms. This is the fungus. This is the fungus. The complete name of this fungus is Penicillium notatum. Penicillium notatum, and this fungus is producing the antibiotic. The antibiotic adds into the solution drop by drop, and then we remove it by filtration, and waste products are separated. So this is the procedure of penicillin production. Any problem?
no,
Next thing is mycoprotein. Remember, the word myco means fungus. The word myco means fungus. Have you ever uh, eaten a mushroom or seen a mushroom?
yeah, both.
Are the mushrooms edible?
Some of them.
Yeah, exactly, some of them. So basically, what's happening that uh, the mycoproteins are the fungi other than mushrooms, and these fungi are edible. These fungi are edible. So basically, we produce these fungi inside the bioreactors so that we you can say this particular fungus Fusarium venenatum, we grow it inside the fermenter, and when we grow it, it contains no cholesterol and lower in saturated fats, suitable as a part of vegan diet. So many people use a particular type of meat called as corn. This is a type of meat, but it is not a meat. But this is basically a protein that is used in replacement of the meat. And this protein is called as the corn. And corn is the protein that is eaten by many of the people that which are vegans. So basically, it is produced in the bioreactors. How is it produced? We grow a huge volume of Fusarium venenatum, venenatum, and when we produce them, we convert their filtered product into blocks. So it's as simple as that. We convert their products, and then we eat them as like it looks like meat, and like this is how the corn looks like these looks like the boneless meat. Now these conditions we often control in the fermenter, as I have already told you. Okay, any problem?
No,
Now we are about to study a whole new topic called as genetic modification or genetic engineering. There is a technology in there is a technique in biotechnology called as recombinant DNA technology. Recombinant DNA technology, basically, we add a piece of DNA, we add a piece of DNA or a gene into a microorganism, into a microorganism, especially bacteria, especially bacteria. Uh, we add a piece of DNA or gene into a microorganism, especially bacteria, and especially bacteria, or I would say bacterial DNA. This process is called as recombination because we have added a new gene called recombination. Since since a foreign piece of DNA has been added, a foreign piece of DNA has been added. So this phenomena is called as recombination. Why do we do it? See, some medicines cannot be made in the lab, cannot be made in the lab, and especially proteins. Some medicines cannot be made in the lab, especially proteins, because we need cellular system for that. Cellular system for that purpose. Or you can say for their production. So what do? We take a gene, we take, or I would say, we isolate a normal gene, we isolate a normal gene. By isolation, I mean we take out a gene, and this gene is sometimes called as gene of interest. Gene of interest. Gene of interest means that the gene for which you need a product, for which we need a product, a product. Okay, so we isolate a normal gene. Now we have a bacterium. We have a bacterium. Remember that this bacterium has to be a plasmid containing bacterium. Plasmid containing bacterium. Why? Because we cannot play with the chromosomal DNA. Chromosomal DNA means it's the primary DNA for the bacterium. The plasmids are non-chromosomal pieces of DNA. These are plasmids. They are non-chromosomal. If you remove a plasmid, if you remove a plasmid from the DNA, I'm sorry, if you remove a plasmid from the bacterium, it would almost have no consequence. Bacteria wouldn't die. But if it does die, we can use another bacteria. But the plasmid, we do not have a problem with bacteria dying. I'm sorry. Like this looks like a very harsh sentence, but we do not have a problem with the bacteria dying. The problem is we we basically need a DNA that is accepting something. Plasmid is a receptive DNA. Like it easily, you can simply say that it easily integrates a foreign DNA. And this is one of the reasons of the bacterial success, when the bacteria, most of the time the viruses attack on the bacteria, and that virus is broken down by the bacterium, like there is a technique in bacteria called as CRISPR. Although we are using that particular technique for our own purposes these days on a very wide scale, but the CRISPR was first discovered in the bacterium. The CRISPR is a technique in which the bacteria simply crush the virus. CRISPR. It's the bacteria simply crush the virus, and when it crushes the virus, it just simply you can say converts it into small DNAs, and out of those small DNAs, the plasmid can intake some parts of it. So it's a receptive. The plasmid is a receptive type of DNA. So we choose the plasmid. Now, what is most important thing? The most important thing in a plasmid, or I'm sorry, the most important thing in a recombinant DNA technology is to isolate a gene. Remember, we don't need a whole piece of DNA. We just need a small piece. We just need this part. We don't need whole DNA. This is the gene we require. So how do we perform such an action in which we can take out a small piece of DNA? Okay, let me give you a small idea out of it. See, when we need a small piece of cloth, do do we start using, like when we try to make a handkerchief out of a piece of cloth? Do we use a whole um, do we use a
No,
whole piece
just
of
a part
cloth?
of it.
Just a part of it, obviously. So when we use just a part of it, it means that we are basically cutting it, and we use it as a handkerchief. Now remember that we do not need to use the whole DNA, we need to use the gene. Now what do? We need some scissor, and that scissors are enzymes, and these enzymes have a very particular name called as restriction enzymes. Restriction enzymes. So we cut the gene with the help of restriction enzymes. Now this is our gene. This is our gene, and then we take out the plasmid, and we treat the plasmid with the same type of restriction enzyme, and the plasmid gets opened. The plasmid gets opened. When the plasmid gets opened, just hold on a second. Okay. When the plasmid gets opened, now we mix our gene and plasmid into the same solution. The new plasmid would now look like this. This is what we call as recombinant DNA. Why? Because we have added a foreign piece of DNA into it. So it is the recombinant DNA. A foreign piece of DNA into it. This is the recombinant DNA. And now what do we need? Next, we need a cell, because remember, no matter how much effort you have put to add the gene into another DNA, you cannot make a protein out of it until you have a cell. So you take another bacterial cell, you take another bacterial cell, and you put this is the chromosomal DNA. This is the chromosomal DNA, and we put the plasmid, just like plasmid is very small DNA. So we put the plasmid into the bacterium. Now, this bacterium is going to be, we grow it. We grow it in bioreactors. This bacterium we are going to grow in the bioreactors, and it can do two things. It can clone our genes. How you suppose that this bacterium would clone our genes? It would produce the protein for us. No doubt, it would protein for us. But how do you think that it is going to clone our gene? Hmm?
Not sure.
The bacteria divides.
Yeah.
The more the bacteria divide, or the more the bacteria multiply, the more copies of our DNA would be generated of our DNA, because like every time the bacteria divides, it create a copy of DNA would be generated. Clone means an exact copy. So it has two benefits. One very particular terminology is that we need is very, it's a very a bit of technical, one of the technical things. Suppose this is our gene of interest and this is our plasmid, and they have been treated with the restriction enzymes. Remember, restriction enzyme has to be same. The enzymes to cut the DNA has to be same both the times. The time when you are cutting the gene of interest and the time when you are cutting the plasmid. One more thing, how would you get the gene of interest? You can take any cell. Let's say you can take a human skin cell, and then you can apply the same rule to it. Take the skin cell, apply the restriction enzyme to it, take out the DNA. Any cell can be taken. So the important part is how the gene of interest, how the gene of interest finds exact spot to attach to the plasmid. This is a big question. Now, to understand this, you need to know that suppose that you have a DNA, you have a piece of DNA, and you treat it with the restriction enzyme, treat it with the restriction enzyme. The restriction enzyme cuts the DNA diagonally, meaning that when the gene is cut, it leaves some pieces, it leaves some pieces like there is no there is no opposite strand for these pieces. Like it cuts the DNA in a pattern where the ends of DNA don't have their opposite, they don't have opposite strand. And always remember that the DNA is only complete when there are opposite strand. So when the restriction enzymes cut the gene, it cuts in a way that one part of the strand does not have an opposite strand, or in other words, we say that they don't have a complement. No complement. You remember the nucleotide complementary rule?
Uh, nucleotide.
The A would always come against the T and G would always come against the
Yeah.
C.
Mm.
This was the complementary rule. But the
ATGC,
restriction
right?
and yes, now the restriction enzyme cuts the DNA in a way that some part of the strand is not having the complementary strand. The same pattern would be followed in the plasmid. Now you can see this part and this part. These parts are without their complements and these parts basically called as sticky ends. Why sticky ends? Because the moment this gene would reach to the sticky end, right at that time it would attach to it. Right at that time, because remember that they are actually complementary. Why they suddenly became complementary? Because the restriction enzyme cut them in a way that they both are having sticky ends. They both are having sticky ends. Sticky ends would be defined as a strand of DNA, a strand or part of gene without its complement. Without its complement. So it would fit into the plasmid, and now it would be simply one more enzyme you need. One more enzyme you need. Let me tell you. Suppose that this is the plasmid, and this is your DNA. Like this is our DNA. Now remember that at the end, when the plasmid and our gene have adjusted, when the plasmid and our gene have adjusted, a small part has left here. You can say this part, um, what do we say? Sewing. Like this part is unstitched. This part is unstitched. Now we need the stitching enzyme. We have matched both the DNAs. Now we need a stitching enzyme. So for the stitching enzymes, we use an enzyme named as ligase. Ligase is the enzyme that stitches both the DNA molecules, and your DNA, recombinant DNA, is ready. You put it into the bacterium and you produce the insulin. This is what they are basically talking about here. Yes, over here. Genetically modified organisms. Understood?
Yes.
Okay, so now we will go through some data. Although everything is reading material here, there is nothing very technical. Everything is reading material, but we do go through some data. Let's see what we have. See, what are the applications here? The applications of genetic modification. Remember that when we talk about the application of genetic modification, we are not restricted to the human proteins now, although we do produce human insulin, and all the people that are able to take the insulin injection, they have to thank the scientists for this particular phenomena they figured out, and they figured this out. But there are other human proteins as well, like there is insulin gene. Other than that, you can just imagine that insulin gene to produce the insulin. Then we use the, like initially it was produced by the taking the pancreas from the pigs and cattle. Initially, when there was the problem of sugar, we used to take the insulin from the pancreases of pigs and cattle, and obviously it was a very riskier procedure. Then this is the humulin. Humulin means human insulin. The hepatitis B, this is going further, we can ignore it. Although we are producing hepatitis B vaccine. How we are producing hepatitis B vaccine? We give the antigen genes to the fungus. Now we are modifying the fungi as well. So we give antigen genes to fungus and it produces antigens for us. We insert the antigens into our bodies and produce the vaccine, produce the antibodies. Then another application is genetically modified crops. We are not limited to bacteria anymore. So we are inserting the genes into the plants. So we can have such plants which are you can simply say we can have such plants which are resistant to certain type of diseases or the bacterium. This is one of the phenomena. The Bacillus thuringiensis. Uh, this produces a toxin that kills the caterpillars or insect larvae. You know what destroys our crops? The thing that destroys the crops mostly are the larvae and caterpillars. Like they are so vigorous eaters that they just you cannot even imagine competing them. Why? Because this is their eating age. The larva means eating age. The caterpillar means eating age. So they eat a lot and they mostly destroy the crops, their leaves, their fruits, everything. So we take a gene from this bacterium called as Bacillus thuringiensis, and we insert the this bacterial gene into plant. Now the tables have turned. Now what we are doing, we are taking the bacterial gene into plant. Previously, we were taking the human or the higher organism gene and we were putting them into uh, bacteria. But now the tables have turned. The gene for the toxin has been successfully introduced into some plant species such as maize, cotton, soybean using bacterial vector. Bacterial vector. What is vector? Okay, do you have idea of vector? Not
You know,
physics. Mhm.
not the physics one, right?
It basically, this word is basically used for a biological vehicle, a vehicle or transport. Let's suppose I can say the plasmid is a vector. Why? Because it carries our genes. It carries our genes. So it's a vector. So next is we this base in this phenomena basically this is very cunning. This bacterium is very cunning. Usually this bacterium is dangerous for plants, but we use a very cunning technique. We neutralize this bacterium. We neutralize this bacterium, and when we neutralize this bacterium, now it cannot cause diseases in plants. In plants. Instead, instead, it transfers, it gives its gene to plants. Basically, this bacterium, why it is dangerous? Because it causes cancer. The When the bacteria is a pathogen, meaning disease causing, it transfers its genes into plants, causing cancer, causing cancer. But when we neutralize it, it cannot cause disease or it cannot cause cancer, but it still transfers the genes. See, this is another way of bacterial infection. Like previously, the bacteria was causing the infection by releasing some toxins. Right now, in this example, we are saying that bacteria is simply transferring its genes into plant so that it can cause cancer. But we neutralize it. Now it does transfer the genes, but not cancerous genes. And those genes, when they get placed into the plants, they do benefit the plant by producing such a toxin that is that protects them from larvae. Unfortunately, there are signs that insects are developing immunity into the toxin. That is another problem. The insect is quickly one of the most quickly adapting group of animals. Quickly adapting, and by adaptation I don't mean adjusting, I mean permanent change in their body phenomena. They bring permanent change. That is why the pesticide companies are so much earning, because in nature the insects are constantly developing immunity against pesticides and they have to create the new pesticides every year, so they are earning much. Then there is herbicide resistant. Okay, what is a herbicide? Obviously herbicide is a chemical that we use to kill herbs. The problem with it, that it destroys our crop plants. So we insert the genes into the plants, we insert the genes into the plants. A gene for an enzyme that breaks down glyphosate is introduced in plant. The glyphosate is the herbicide. The glyphosate is a herbicide. So we inserted the gene into the crop plants. Remember, we are saying about the crop plants. We inserted the gene into the crop plants so that the crop plants can build a resistance against the glyphosate. And it is said that maybe it's carcinogenic. Modifying plant products means that we modify them for our food. Like we develop the golden rice for more vitamin A levels. We modify the maize to increase iron uptake so that when we eat the maize or corn, we get more iron. We modify the wheat to make it free of gluten, which is a major cause of food allergy. Vegetables with a gene inserted to boost vitamin E. There are vegetables that have vitamin E in it. Cassava with genes inserted to increase root iron uptake, so it contains more iron. Now, there are advantages and disadvantages. The problem is that in advantages and disadvantages, these are really simple. This is something very simple data. So, what are the possible hazards and like what are the possible advantages and disadvantages? So, what do you say? We move to the next chapter. Can you read this on your own?
Yeah.
Because I guess for 10 to 15 minutes, I'm only reading the data. So we can move to the next chapter. We can cover up some basics of it so that we can continue and finish the next one, because you can say luckily or unluckily the next one is again too much reading. Next one is again too much reading. Only the first chapter of it is the only the first chapter of it is actually the conceptual. So let's see. Let's start the next chapter. The next chapter is the last chapter is this one: organisms and their environment, and then the food chains and food webs. This has been divided into many components. Each and every small line has been divided into a concept, basically. That is why, like, some of these are primary level concepts, the primary classes. Okay, first of all, let's talk about ecosystem. What do you know about an ecosystem?
Um, Habitat for animals, or
Okay, any more
anything,
explanation? Hmm?
anything where like different organisms work together to build an
Yes, you are very
environment.
close. Mhm, mhm, mhm. Basically, the ecosystem is a group of organisms belonging to different species, different species at a particular location, at a particular location. This basically is the definition of community as well. A group of organisms or a community where organisms interact interact within themselves. No, sorry, where organisms interact with biotic and abiotic factors of their surroundings, of their surroundings. Now remember that the ecosystem is all about interaction. Ecosystem is all about interaction, meaning that in an ecosystem, the organisms are interacting with each other. I'm not saying interaction is always positive. I'm not saying that interaction is like it is always going to be very much productive, but it is an interaction. Still, it is an interaction. See, obviously, we interact with our colleagues, with our peer group, and what happens that sometime we don't agree with them, sometime we get into a fight even. So still it's an interaction, no doubt it is a negative one, but it's an interaction. Similarly, an ecosystem is overall made of interactions. So remember that the in an ecosystem we have two kinds of factors that are written over here. One is called as abiotic and other one is called as biotic factor. So let's see what are biotic factors of an ecosystem. Can you tell me what are the biotic factors?
Uh, like plants, animals,
All
microorganisms.
living things in an area or an ecosystem. Like the biotic factors are like you can name them or you can just shorten their definition by just saying that they are the living things of an area. So these are the biotic factors. Okay, I will discuss. Obviously, by abiotic factors, you can easily determine that an abiotic factor is the one which includes all the non-living objects. It includes all the non-living objects in an ecosystem. What are their examples? We'll discuss that later. Right now, let's talk about the biotic factors. See, in the biotic factors, we include all the living organisms. But since we are targeting the relationships, or interactions in an ecosystem, or interactions in an ecosystem. So first of all, we need to make the subgroups of biotic factors. Otherwise, we cannot understand what are the interactions. So first of all, let's divide the biotic factors into the subcategories. Um, in biotic factors, first of all, we include those organisms which make their own food. You can say, okay, hold on. Basically, we can further classify, we can further classify, what happened? Did I
Uh,
leave or did you leave?
I got disconnected. Um,
Okay, what was the last thing you heard?
you were right, starting to write relationships.
Okay, basically, I was saying that if we are understanding biotic factors, if we need to understand the best, because uh, understanding the biotic factor is not that much of a problem because they are all living things. But our main thing to understand is the relationship and the interactions in an ecosystem. So, what do we need to do? We first need to understand that what kind of relationships they are in. What kind of relationships they are in. So for that understanding, we further classify the biotic factors, and those biotic factors, when we further classify them, our criteria is mode of nutrition. And I guess you remember that I told you there are two different modes of nutrition. One is called as the autotrophic, and the other one is called as the heterotrophic. Okay, the autotrophic mode of nutrition, on the basis of autotrophic mode of nutrition, we name the group of biotic factors as producers. Obviously, what are autotrophs? What are autotrophs?
Autotrophs, so like the organisms that can produce their own food.
Hmm. Photosynthesizing organisms, obviously.
Yeah.
So they are autotrophs. So obviously they can be plants only. They can be plants only, or maybe other type that follows the same procedure. So we categorize them into the producers. We categorize them into the producers. The producers are what kind of production they do. If we name them producers, then what kind of production they are doing? Can you guess?
If they are,
If they are producers, then what kind of production they are doing?
they're producing food.
Food, or can you name it something else?
Photosynthesizing.
No. The production that is done by the producers is energy. They are producing energy, or in other words, they are producing the biomass, mass of life. So basically, why do we call them producers? They do produce the food, no doubt. I'm not contradicting your that statement. I said, what else, what other name we can give that production? So they are called producers because they are producing the energy. Okay. In other way, we can say that what do all organisms require to remain alive? All organisms require to live. The answer is same, energy. So you can say the producers are basically the energy source for an ecosystem. They are energy source for an ecosystem since they are producing the energy for them. Got it?
Yes.
Next, the heterotrophs. Now, the heterotrophs, since they cannot make their own food, they go to the producers for the food. So we say that they are the consumers. Consumers of what?
Of food,
Again, what else? What other name?
energy.
Energy. They are the consumers of energy. So basically, we have divided them into two groups. But the problem is there are many types of consumers. So what do? We divide we divide the autotroph and heterotroph into what? Into trophic levels. Trophic levels of an ecosystem. Trophic level mean nutrition level or energy level. So the plants are at T1, meaning that they are at the first trophic level. And the heterotrophs, coming to the consumers. Now we further classify the consumers because not all consumers, not all consumers take energy directly from plants. Not all of them take energy directly from plants. Some take the energy from the plants, A, then the A gets eaten by B, so the energy transfer from A to B, then to C. So you can say that you can say that actually the energy is getting transferred. The energy is getting transferred to the consumers, but the consumers are further divided according to their, like you can say, uh, mode of obtaining energy or the level of energy. Like the trophic level 1 represents that they are the producers. The A1, the A organisms, the consumers, these are called as primary consumers. Primary consumers are those which are getting energy directly from source. Getting energy from source. So they are primary. Then obviously T3. They are at the third trophic level. Obviously they are heterotrophs. Remember, below this all are heterotrophs. There is no difference between them being a heterotroph. The only difference is what source they are using. Whether it's a direct source or an indirect source. If they are using the direct source, they would be called as primary consumers, and they are considered to be at trophic level 2, or you can say energy level 2. The trophic level can also be named as nutrition level or energy level. So then C, the energy would again be transported to the next level. This is called as T4. You can simply say the chain of or you can say, linear chain, linear chain of energy transfer is called as food chain. And most organisms, most organisms are in energy relationship, or you can say most biotic factors are in energy relationships with each other. Most biotic factors, let's say, let me tell you, uh, there is a predator prey relationship.
Yeah.
There is a predator prey relationship. Why that relationship exists?
I think for population management also.
That is something else. But in reality, an ecosystem, the predator prey relationship exists. The predator prey relationship exists because in that relationship, the organisms one organism is getting energy from another. So one is at trophic level 1 and the other one is at trophic level 2. What about the cheetah and a deer? The deer is at trophic level 1 because it's a primary consumer, while the cheetah is at trophic level 2 since it's a secondary consumer. So always remember that ecosystem, in every ecosystem, the the trophic levels represent the energy level. And if the relationships exist in an ecosystem, they basically exist for the transfer of energy, for the transfer of energy. So every time we need to remember that the transfer of energy is the real thing. The transfer of energy is the real thing. And in all types of ecosystem, all types of ecosystem, the primary type of relationship between different kinds of organisms or trophic levels is the energy relationship. Just hold on a second. So technically, we can say that all types of relationships in an ecosystem between the biotic factors is actually related to the energy. Whether you say it's a predator prey relationship, whether you say it's a competition, you may say that it's a symbiotic relationship, a kind of relationship in which two organisms live according to mutual understanding. Every relationship is energy based. So then trophic level 3, then trophic level 4, the energy gets on transferred. So when we divide them according to according to the mode of nutrition, we must also remember that the producers have the main source of energy as the sun. Obviously, the photosynthesis occurs under the in the sunlight. Then when it goes to the consumers, we start dividing them. And then the food chain is built. Food chain is a linear chain of energy transport. So this is how we divide them. Then these are the secondary consumers, then tertiary consumers. Now there may be another terminology you may have heard in your junior classes, and that terminology is the carnivore, herbivore, etc. So remember that those terminologies are not technical terminologies for an ecosystem. For an ecosystem, herbivore is nothing. It's a primary consumer since it eats the grass or the green food. So it's a primary consumer. The herbivore is even a herbivore is a mistaken name. A carnivore is sometimes a mistaken name. We say the dog is a carnivore. But you can see the people who pet the dog, they give the dog, the fruits, the vegetables, different types of other salad vegetables. So what is it? Is it a carnivore? It it can be put into the omnivore. That is why these terminologies are misleading. So technically, when we study the ecosystem, an ecosystem apparently at all levels or at the levels of IGCSE, it's very simple. But in reality, when it comes to the ecosystem, it becomes very difficult. It's extremely simple here in this at this level, because when we come to the energy relationships between the organisms and we try to calculate the total energy production by the organisms, we come up with with very difficult situation. So in reality, in ecosystem, the herbivores and carnivores are not the biotic factors. It's the primary consumer, secondary consumer, and tertiary consumers, etc. Got it? Any problem?
No, understood.
Now, when we come to the ecosystem, what is our main focus? Our main focus is energy transfer, and the energy changes. These are very important to know. This is very important to know that energy changes and energy gets transferred through food chain. The amount of energy changes when it transferred to the next trophic levels, the next nutrition level, or the next food level. The energy changes and how it transfers. How do we represent the energy changes? That is going to be our very next topic, representation. All right.
Yeah.